Hello, good afternoon. Let's start with a little bit of a review of where we left off last time. It shouldn't take too long to finish the rest of the slides in lecture 6. And then we're going to start on lecture 7 today as well. So last time we were talking about the cell cycle. Right? So here is our cell cycle. And the cell spends most of its time in what we call the interphase. Okay? The interphase is here. And that's just the cell performing its normal function, uh, growing, gathering resources to prepare for the actual division, the mitosis. Okay, so most of the time is spent in this interphase. And within interphase, we have the G1, we have the G2, and then we have the S phase. That is basically different points where you can check the quality of the DNA and stuff like that. So we call this like checkpoint number 1. And this is like checkpoint number 2. And the S phase here is the synthesis, where you actually going to be copying the DNA. So before you copy the DNA, you want to check to see if the DNA quality is good, any mistakes, any errors, if the telomeres length are long enough to go through another round of division, and if you have enough nutrients to go ahead with the division. And if everything is good, then the cell is given the green light, and they're going to go ahead and copy the DNA. And afterwards, you also have to check for the quality of the DNA that you have copied. Right? Did you make any mistakes during the copying? Were you able to copy everything, and so on and so forth, right? So there are these checkpoints that are in the interphase to make sure everything is done properly, right, before you allow the cell to go ahead and do the mitosis. Now, these checkpoints are carried out by various types of proteins, and we learned about 2 kinds of proteins. Those that are going to be called growth factors, and they are responsible for promoting cell division. The green lights, I call them, right? And then we have the growth inhibitor. And these are going to stop cell division. Okay? And these are proteins that are going to be doing the job. And as you know, proteins, the instruction to make the proteins are encoded within the DNA. And so the gene that is going to be giving the instructions to make these growth factors, they are called the proto-oncogene. And what happens is, if you have a gain of function mutation, that is going to turn the proto-oncogene into a bad gene. And the name of the bad gene is an oncogene. And gain of function means it's always going to be turned on, it's always going to be green light. And as such, the cell will be able to divide continuously. On the other hand, we have the growth inhibitors, which are coded by tumor suppressor gene, which, when we have a loss of function mutation, then it's going to be always off. Okay, we don't have a special name for it. It's still tumor suppressor gene afterwards, but it's mutated. It's not working anymore. And when the red lights are always off, We can say this one is always on. So when it's always off, then it's basically not able to stop the cell division. Okay. I also mentioned how here, for this to have problem, you only need one hit. Right? If one of the green lights is always on, that's enough to cause the cell to divide uncontrollably. Whereas over here, these are chromosomes, by the way, you need to have two hits in the same cell. Right? Both of the red lights have to be not turning on for this to be a problem. Okay, that's where we left off last time. Does anybody have any question about any of these before I continue with today's lesson? Okay. So when you have accumulated enough mutations, like in a real life situation, it's not just gonna be a single, growth factor mutation or, tumor suppressor gene mutation. It's like usually a bunch of these things, like many red lights are not working, or there are many green lights that are always on. Then it leads to uncontrolled cell division. And when the cell keeps on dividing and dividing, you end up having a tumor. And a tumor is basically a collection, a lump of cells, right? And we have a biology term for a tumor. It's called a neoplasm. Okay, neo means new, right? It's a new mass of cell, basically. Now, tumor is not necessarily cancerous. I told you that last class. Tumors can be benign or they could be malignant. Benign is the harmless version. It's just a collection of cells. If you remove it, usually they don't come back and there's no further complication and life goes on. But if it's malignant, then it could get progressively worse. Surgery can remove most of it, but it might come back. And that's the cancerous version of the tumor. So nobody wants a malignant tumor. A benign one is okay. But how do you know if it's malignant or benign, right? Well, one of the things that people do when there is a tumor is they will go have a biopsy done. Right? So a biopsy is basically like the doctor will take a sample of the tumor and then they will do tests on it. Okay, they can look at it under the microscope. They can run it through different instruments, and they will identify markers or characteristics that is associated with cancer. Okay, so we're going to look at 6 of these characteristics. But generally, cancer is bad news, right? Like here you have a normal cell, and there is some kind of initial mutation that happens. Mutations can be spontaneous, which means it's just bad luck. It could happen to anybody. The mistake was introduced when the cell is like copying its DNA, for example. It's just made a mistake. The mistake was not caught and it was not corrected and it stays there. So that's a spontaneous mutation. The other kind of mutation is induced. Induced meaning it is exposure to carcinogens, right? Carcinogens or mutagens, right? These are things that would cause changes in the DNA. Could be things that are found in cigarette smoke. Could be chemicals that are found in even everyday life products, right? People say, your Teflon coating, the non-stick pan stuff, that stuff, if you heat it up too high and then it ends up being in your food and you eat too much of it, that's linked to cancer, increased risk of cancer as well. So these are induced mutation. It's something that is not happening by chance, but by exposure to things in our daily lives. And then once that process gets started, once the initial mutation happens, then they tend to accumulate more and more and more mutations as time goes on. Because imagine this, right? Like I already told you about the tumor suppressor gene, which can code for a bunch of things. But let's say we have tumor suppressor gene that checks for DNA damage. Okay, the function of this protein from the tumor suppressor gene is that it checks for DNA damage. Let's say that stopped working because of the initial mutation, right? So what happens is in this particular cell, the DNA will not be checked anymore from that point on. If no one's checking the DNA, then it makes sense for that particular cell to end up getting more and more and more mutations, right? And these mutations can make it easier for the cell to keep on dividing and dividing, right? So as the cancer progresses, it's not uncommon for it to get even more mutation, and then it just becomes very aggressive in terms of growing. And at very late stages, some cancer can spread. We call that metastasis. And many forms of cancers are still incurable, and death is the unfortunate outcome. So what are some of the characteristics of cancer cells? Well, scientists have identified 6 features. We call them the hallmarks of cancer cells. So when they do the biopsy on the tumor and they see these things, then that's how they make the decision. Okay, this one is malignant, is cancerous, as opposed to benign. And of course, the outcome of the biopsy is going to determine what you need to do to try to as a treatment option, right? So let's take a closer look at these hallmarks. First 2, right, is number 1, self-sufficiency in growth signal. Okay? They don't even need growth factors to tell them to divide. Because of mutations, because of the mutations of the proto-oncogenes into oncogenes, they have their own growth signal. They have their own green lights that are always on. Okay, so they have their own self-promoting signals. They don't need to rely on other signals to tell them to divide. They will just go on their own. Number 2, they are insensitive to anti-growth signal. Even those signals are telling them to stop, they don't respond. Right? They no longer care. Right? There's a red light, they don't care. They just keep on going and dividing. So these 2 things that are combined together makes the cell very aggressive in terms of division. Okay? Again, they don't need someone to tell them to divide. They would just do that on their own, using their own signal. And if you try to tell them to stop, they don't listen. They don't care. They just keep going and divide. Now, all this division, all this cellular activity actually requires a lot of energy and resources. So another characteristic, and I'm not going in the order that's on the slide, but it makes sense to talk about this one, right? Is sustained angiogenesis. Genesis is to creation, and angio is blood vessel. Angiogenesis is basically creating new blood vessels. Okay, like I said, it's a lot of nutrients that are needed to sustain all this cell division. And the tumor, the cancerous tumor is able to create its own network of blood supply. Okay, it taps into the main blood vessels that are nearby, and then it causes it to grow some new ones into the tumor, and basically suck the nutrients from the nearby cells, right? And then your cells end up not getting the nutrients, and it's like this competition for limited resources, and often the cancer wins. And the cancer cells can evade apoptosis. They can cheat death. Right? When a cell is rapidly dividing, usually we don't like that. We tell them to stop and trigger apoptosis to kill it off. But because of the mutation it has accumulated, it essentially ignores that. Okay, it becomes immortal. Lastly, they have limitless replicative potential, partly because of the telomerase. That's a turn on in the cancer. Okay? Without the telomerase, the telomeres get shorter and shorter and shorter. And when it's critically short, the cell no longer divides, right? It becomes senescent, right? It becomes old, basically, and it stays that way and won't divide further. However, in cancer, because of the telomerase, you're able to reset the length of the telomeres, and they no longer become senescent. They no longer become old. They just keep on dividing. I know I keep on repeating the same thing, but that's what it is. Cancer are cells that won't stop dividing, and there are many factors that allows it to do it. And finally, at advanced stages of cancer, essentially the tumor, the cancer cells, they grown too big, and they've got all these blood vessels going, and they basically suck dry the nutrients at this particular location. So the only way for them to keep on growing is to invade other tissues. Right? So some of it could break off, travel through the bloodstream, find a new site and attach, and start a new site basically, and that's called metastasis. Okay, so 6 hallmarks of cancer: self-sufficiency in growth signal. It can have its own green light. They ignore inhibition signal. Okay? They can evade death, evade apoptosis. They can create their own blood supply. They can keep on dividing without becoming senescent, without becoming old. And when all the resources at this location is exhausted, they could break off and then find a new location and grow. So there you have it. That is the 6 hallmarks of cancer. Here are a few multiple choice questions that I would like to go through with you. You might remember the idea of penetrance. The penetrance is this concept that talks about, if you have a particular gene, how likely are you going to be expressing that gene? Okay, and it doesn't have to be a bad gene. Okay, but in the context of what we are talking about, we are talking about bad genes, like a cancerous gene. But how likely are you going to get cancer, right? So the answer for this is C. Okay, low penetrance means even if you have this particular gene, it's not guaranteed you're going to get the disease. Is when you have the complete penetrance, then getting a particular gene guarantees you going to have the condition. Okay, so that's the answer is C. A proto-oncogene, I just reviewed that with you earlier today. A proto-oncogene is a normal gene that involves in promoting cell growth. Okay, it's the green light, right? It does not cause cancer. It only causes cancer when it mutates. Once it mutates, once you have like a gain of function mutation, then we no longer call it a proto-oncogene. What do we call it instead? An oncogene. The oncogene is the bad one. Which of the following is not a tumor suppressor gene? Okay, tumor suppressor gene has everything to do with slowing down the growth. All right, so when do you want to slow down the growth? When you have to do DNA repairing. Okay, so that's a tumor suppressor gene. If it's not supposed to grow, you can trigger apoptosis, cause it to die. That's a tumor suppressor gene. Accelerating cell cycle, no, that's not a tumor suppressor gene. That would be the function of a proto-oncogene. And then checking for error as well, that would slow down growth. So the answer is C. And I think that's it. That is it for lecture 6. Now, does anybody want to ask me anything about lecture 6 before we do number 7? Number 7 is pretty intense. There's a lot of stuff that we have to cover and discuss. So I just want to make sure that you guys are okay with 6. Yes, Blessing.
Yes. Last class you said cancer is not hereditary, like it's not transferable from generation to generation. that is caused by environmental factors, right?
What is not transfer from generation to generation? Sorry, I missed the first part.
Like this cancer cell
Oh,
is
cancer
not
cell. Okay, yeah.
is
Okay, yeah.
not yes. So what
So,
are the factors that causes it?
okay. So, good question. Now, the cancer is at the root cause of it is basically mutations, right? Okay. So some mutations, if it happens spontaneously, okay, and it happens to be in the gametes, like if it happens in like the sperm and the egg, then it's possible for you to pass it on to the next generation. Okay, but if it's something that you acquire, like if the person acquires it during their lifetime, let's say the person smokes a lot, okay, and then they get the lung cancer from the smoking, that's not going to pass down to the next generation, right? But however, like the one that we talk about, do you remember the BRCA1 gene? The BRCA1, the breast cancer gene, that's a tumor suppressor, right? So if that's something that's already in the genome, then there's a chance for you to pass it on to the next generation, right? Because like it's not something that you get in your life, right? It's already there. You inherit it from the previous generation. So anything that any mutations that you're born with because you inherited from the previous generations, those are the kind that would get passed on to the next generation. And when it does, it doesn't mean the next generation is going to get cancer, but they are more likely to get the cancer, right? I don't know if that answers your question.
I
But
see.
yeah, so basically things that we acquire throughout our lifetime, those are not going to affect the future generation. Okay, like if I like to spend a lot of time outdoors in the sun and I don't put on, sunscreen, I expose myself to UV. Those UV could damage my DNA. As long as it doesn't damage the DNA in my gametes, right, or my germ cells, that's not going to pass on. Does it make sense?
Perfectly. Thank you.
Okay, great. All right, number seven. Number seven is about protein synthesis. All right, so I hope you have the notes with you because there are some example questions we have to do together. And it's an important lesson because it paves the way for a lot of stuff that we will continue to learn in the course. So I just really want to make sure that we have a good foundation here in this particular lecture. So we want to make proteins for many reasons. We have tons of proteins in the body. You should know that by now, right? Well, hair is made of proteins, muscle is made of protein, and we just learned that these checkpoints are made of proteins, protein everywhere. But of course, the protein, the instruction to make them is in the DNA, right? And we're going to make the DNA, make a copy of the DNA in the form of messenger RNA. And then we are going to use the copy to make protein, right? You might remember from DNA to RNA, that's called transcription. And then from RNA to protein, that's called translation. So our job today, my job today, is to teach you how to decode the DNA, how to change the ACTGs into proteins. Right? Proteins are amino acids, right? So how do you know what code corresponds to what protein? That's the key thing to learn for today. If you have learned this before, great. If it's the first time, ask questions, okay? It's complicated. I try my best to explain, but if you don't get it, it's okay. Just ask. I'll keep on explaining it in different ways until you get it. Okay, you have to get it before you go today. So let's start with transcription. Some terms that we have to remember, right? When we discuss transcription and translation. First of all of your genetic material, we call that genome. Genome. That's all of your genetic material. Chromosome 1 all the way to chromosome number 23. Okay, that's a genome. So if you think about I don't have a textbook here, but like if you think about a whole textbook, the whole textbook is like your genome. Okay, and then each chapter is corresponding to a chromosome. Okay, so we have like 23 pairs of chromosomes, right? So chapter 1, that's chromosome 1. Chapter 2, that's chromosome 2, so on and so forth. And then you have the chapter 23, that's chromosome 23. Okay, so like that's the book. Now, a gene is a section on the chromosome. Right? So like if we look at chapter 4 of the textbook, right? Under chapter 4, inside chapter 4, I should say, there's like section 4.1, 4.2, 4.3, right? And each section talks about a specific topic, right? That's what a gene is. A gene is a section, a segment of DNA on the chromosome that gives you the instruction to make the protein. So there's a gene A here that gives you the instruction to make, protein A. Then you have gene B and gene C and gene D. In reality, though, it's not just 4 genes per chromosome. You have hundreds and hundreds of genes on each chromosome. Okay? The location of the gene, right? Like where are you gonna find gene B on the chromosome? Okay? That's called the locus. So locus is like the page number in the textbook. All right, like I want to find section 5.6. All right, where is it? What page is it on? I want to find gene F, okay, on chromosome 9. What's the locus? What's the location? Are we together on genome, gene, and locus? Okay, now next slide. We have 23 pairs of chromosomes. You should know that by now. I've been like repeating this message, forever. 23 from mom, 23 from dad. And so if you line up the chromosome, there I show you a real picture of chromosome. Okay, this is called a karyotype. We learned that in more detail later. But karyotype basically showed you all the chromosomes. Okay, so like this is chromosome 1. This one is just curled up. Sometimes they curled up. Okay, like you can straighten it if you want. So that's chromosome 1, chromosome 2 and 2, 3 and 3, 4 and 4. So on and so forth, all the way up to maybe this is not the best picture because it doesn't show all of them, but you know better, right? It's gonna go chromosome 21 and then 22, 23, right? My point is the chromosomes are like a pair of shoes, okay? Like if you have, I don't know how many pairs of shoes you have, but let's say you have 23 pairs of shoes and I randomly throw them out on the floor, you would have no problem matching them up, right? Like you pick up one shoe, you're like, okay, I know exactly what the other one looked like, and then you put them together and you get your pair back, right? That's exactly the same thing for geneticists. If you mix up the chromosome 1 to 23 or whatever, right? They have no problem finding, like they can group them together. They can find, okay, chromosome 1 with another chromosome 1, chromosome 2 with another chromosome 2. Do you understand that? Okay, so back to the shoe analogy, the left shoe and the right shoe, when they are in a pair, right, one from mom, one from dad, we call that homologous chromosome. Homologous chromosome. Okay, so chromosome 1 is homologous to chromosome 1 only. Chromosome 7 is homologous to the other chromosome 7. Do you understand? Right? Think of the 23 pairs of shoes. Right? Each shoe is homologous to the other half of the shoe only. Are we together on this? Please let me know if you if you need me to explain this further. For real? I'm doing such an awesome job that all of you understand the homologous chromosome? All right. Now, the homologous chromosomes are same length, like 1 is the same length as 1. Okay, and then 2 is a little bit shorter than 1, 3 is a little bit shorter, and so on and so forth. So the homologous chromosome is same length, and the order of genetic information is the same on the homologous chromosome. If you find gene A, B, C, D on one chromosome, you would expect to find them in the same order and at the same locus, same location on the other chromosome. Okay? Think of the book analogy again. All right, you have your whole textbook, right? So let's say you have two of the same textbooks. Okay, one is in English, one is in French. Right? It's a slightly different information, but if it's the same textbook, you would expect to find section 4.2 on the same page, right? It's just that the way that it's written is a little bit different, right? And that's what happens with these genes. Even though they have the same gene, but the version of the gene might be different. They don't have to be different, but they could be different. So like if you look at gene A here, they are both capital A. That means they're the same version, right? Perhaps both your mother and your father have brown eyes. That's why the gene for eyes is brown eyes and brown eyes. They could be different if we are talking about like blood type. Right? So maybe your mother has type B blood, maybe your father has type O blood, then the version of the gene that you get, they are still both blood type, but it's different version of it, right? Okay, so we call these different versions alleles. So in the population, there could be many different alleles for the same gene. Just take eye color, for example, right? It's not just brown eyes and blue eyes, right? What other eye color are there? Green eyes, right? Like hazel eyes, right? All these different colors are different alleles that exist in the population, right? They are different versions of the gene. Any question for me? No, for real? Okay, so that's that. This is the central dogma of biology. Again, I first introduced this to you all the way back in lecture 3. Let's just quickly revisit it, right? We already know that we have the DNA and we need to change it to messenger RNA, and then we can change it into proteins. So that's old news. But there's one additional piece of information here that we have not discussed previously, and is this thing called the pre-mRNA. It turns out when we copy the DNA, the thing that we get is not quite ready yet. We can't take that out of the nucleus just yet. So it's called a pre-mRNA, and we have to edit the pre-mRNA. We have to modify the pre-mRNA before it is ready. So that there's just an extra step, and we'll learn what that extra step is in a bit. So let's review the purpose of transcription. Why don't we just take the DNA and use it as is? Why do we need to go through this extra step of having an mRNA? Well, DNA is really, really big, and they are not going to be able to leave the nucleus. Okay? The pores are really small. DNA won't fit. If you think about mitosis, which we learned last class, in order for the DNA to come out, you actually have to break down the nuclear envelope, right? So, it's really big. They're not going to be able to come out. And we don't want them to come out anyways. If the DNA comes out, they might degrade, they might break down in the cytoplasm. We cannot allow that to happen. So that's why we are just going to copy the gene that we need, right? Think about, again, the textbook. right? If you have a textbook in the library and you want to make some copies, you're not gonna photocopy the whole textbook. Number one, that's expensive, right? And it's the same thing for the cell. If you copy all the DNA, it's energy demanding, it's a waste of resources, right? It's expensive, right? So we are not going to do that. You're going to only photocopy the pages that you need, right? Same thing. We are only going to make a copy of the gene that we need. Right? If we want to make protein number 7, then we're going to copy gene number 7 only, and the rest we just leave them be. So that also adds a level of control, right, to decide what genes to turn on and what genes to turn off. You have to remember every single cell in your body, except for the sperm and the egg, okay? Every single cell in the body that's not a sperm or egg, they have the same set of genetic information. It's the same 46 chromosomes that are found in all your bodily cells, in your liver cells, in your heart cells, in your brain cells, in your kidney cells, all the same. What makes them different, what makes a kidney cell different from a liver cell, is the set of genes that get turned on, okay? Even though they all have the same set of cells, but we don't turn on all of these genes in every single cell. That would make the cells identical in the body, right? We only turn on the set of genes that are specific to the functions of the cell. If we are talking about liver cell, it does a lot of detoxification. So we have a set of genes that are necessary for that. We turn them on, okay? Liver cells, they don't really contract like the muscles do. So there's no need to turn on genes that are related to contraction. Do you understand? If we go to the brain cells have other set of genes that needs to be turned on. Everything else that we don't need, we are not doing detoxification in the brain. No need to turn on genes for detoxification in the brain. We leave them off. But then we turn on things that are required for, like, information processing, for communication. Okay? That's why we need to do the transcription. You only copy what you need, not everything. Right? Number 2, second concept to review. Whenever we build new strands of DNA or new strands of RNA, because of the way that the enzyme works, because of the way that the polymerase works, you have to follow the base pairing rule. Okay, the polymerase, when we learn the DNA replication, right, think about it, is not able to copy the strand exactly. What do I mean by that? For example, if you see a strand of AATCCG or something, if the polymerase comes and then it try to build using this strand, what is the outcome? What are you gonna end up building? If you have polymerase 3 that attaches onto this, and then you're building a new strand, what is the sequence on the new strand that you're building? Please tell me. You can type in the chat box if you want. That's the question I'm asking. What is the sequence created by the polymerase? If I have an AATCCG here and the polymerase comes along and build a strand, what is the sequence on the new strand? Anybody at all? We are not moving on until someone tells me the answer. I know some of you know it. Is it because you don't understand my question, or is it because you don't know the answer? Which one is it? So look, this is one strand of the DNA, and then the polymerase, that's the enzyme that builds the DNA, right? Comes along, and then it sees an A. What is it gonna make? That's my question for you. What's gonna be over here? Okay, Sebastian says it's gonna be TTAGGC. That's correct. Does the rest of you understand this? If not, you have to speak up. Otherwise, the rest of the lesson is gonna be like alien language. Okay. Now, my point is this, and thank you, Sebastian, for giving me the answer. My point is this: the way that the polymerase work is when they build a new strand, they have to use the base pairing rule. That's the only way that they know how to work. It's not possible. It's not possible for them to just copy exactly as it is. This doesn't work, okay? The only way for them to work is to follow the base pairing rule. And that's true for all polymerases, okay? Keep that in mind. So what are the base pairing rule? Well, A goes with T and G goes with C. But there are base pairing rules for DNA and RNA as well. And that's going to be useful when we try to do transcription. Because in transcription, you are not making DNA, right? You are making RNA. So the rules are very similar. Instead of A pairing with T, we have A pairing with the uracil. Because there are no T in DNA in RNA, right? Instead of thymine, we have uracil in the RNA. All right. I'm gonna skip all this first, and then we're gonna come back to it because this morning I teach it in this particular order, and people are like super confused. I'm gonna come back to it. I'm just gonna go ahead and tell you about the transcription process first, and then we're gonna do some example. Okay, so we talk about the purpose of transcription, right? We just want to copy a segment of the DNA, the one that contains the instruction to make the proteins that I want at this specific moment. Transcription, of course, takes place in the nucleus. And because we are building the RNA, we are making RNA, it makes sense for us to have an enzyme that's called RNA polymerase. Okay, RNA polymerase. So RNA polymerase is responsible for using the DNA strand to create a single RNA strand. Now, the product that we make is not perfect. I told you it's not ready yet. We call that the pre-mRNA. We must further process it before it can leave the nucleus. Don't worry about the further processing at this point. Just focus on how we make the pre-mRNA. So there are 3 stages in transcription. We have initiation, which basically means the start of the transcription. Okay. And then elongation is the actual RNA building. And then termination is like stop of transcription. Okay. It's really simple. You start, you make it, and then you stop. Okay. Initiation, elongation, termination. Okay. So this I don't know what shape that is, oval teardrop looking shape. That's the enzyme. That's the RNA polymerase. Okay. Remember when we wanted to copy DNA, we had a DNA polymerase. Now we are building RNA. That's why it's called RNA polymerase. Okay, and then we're going to be making the RNA shown here in red, and then when it's done, everything falls off, and then you have your RNA. So let's take a closer look. How do you know where the gene begins? It's all ACTGs, right? In the entire chromosome. Like, how do you know where one gene begins and where one ends? Well, it turns out the beginning of a gene is a special sequence called the promoter sequence. Okay? So in this picture, the promoter marks the beginning of a gene. Just like, you know, when you read the chapter, it says section 5.4, and then there might be like a nice picture there that shows you some picture that's relevant to the topic. You know, that's the beginning of the section, beginning of the chapter. Promoter. The promoter is rich in A's and T's. There's a lot of adenine and thymines in it. And it's not surprising. I've been trying to tell you this since lecture 2. Adenine and thymine only has 2 hydrogen bonds connecting them. So if the promoter has a lot of A's and T's in it, it's easy to open it. It's easy to unzip it. And we have to unzip it. The RNA polymerase has to unzip it to gain access to the DNA information, right? So RNA polymerase will bind to the promoter, this AT-rich sequence. It will unzip the DNA and basically start the process of transcription. It's like, okay, let's start copying here. This is the beginning of the gene. Now, this is the part where you have to pay extra attention to try to understand. There are 2 strands in the DNA, right? 2 strands in the DNA. Which strand are we going to be copying? Which strand do we want the messenger RNA to be identical to? Well, it turns out one of the strand is called the coding strand. Right here, it says coding strand. Coding strand is the strand of DNA that contains the instruction to make protein. That's the actual instruction to make the proteins. And then the other strand right here, that's called the template strand. The template strand has no instruction, does not contain instruction to make protein. So why do we need the template strand if it doesn't contain any instruction? Well, the template strand, as the name implies, is the strand that the enzyme is going to be using to build the RNA. So it does not contain instruction, but it, as in like this, as in a template strand, is used to build mRNA. I told you a couple slides ago, the enzymes can only build using base pairing rule. So if you want to create something that's same as the coding strand, you have to be using the template strand to make it. Right? So like the enzyme, take a look here and say, oh, okay, I see a T. Using base pairing rule, I know I have to put an A here. I see an A, and A pairs with T. However, we are making RNA. That's why we need to put a U, right? G pairs with C, G pairs with C, T pairs with A, and so on and so forth. And afterwards, you will get the messenger RNA, the red one, and take a closer look here. It contains the same information as the coding strand on top, right? So C, C, A, T is replaced by U, but it's the same information, right? And then CC, CC, A. Okay? So, to summarize, the coding strand is the one that has the instruction to make the protein, which means I want my RNA to have the same information as the coding strand. However, I cannot use the coding strand to make a copy of the coding strand. If I did that, the outcome would be backwards, right? Because of how the enzyme works. The enzyme cannot copy exactly as is. The enzyme only works by using base pairing rule. That's why I need to use the other strand, the non-coding strand, the template strand. And when I use the template strand to build my RNA, I end up having a strand that contains the same information as the coding. I'm going to pause there for a second and just let you think about that. And again, if you have questions, please ask. Gotta take off my jacket. All these RNA, DNA talks is making me sweaty. Anything at all before I move on? All right. So that is basically elongation. The RNA polymerase will unzip the DNA ahead, right? Continue to build the DNA, and then as it's moving forward, it will zip up the DNA behind. Okay, so that way you only have a very small bubble as you are doing the elongation, as you are building the RNA. When do you stop? You stop when you reach the terminator sequence. Right? The beginning was marked by the promoter sequence. The end is marked by the terminator sequence. And when you get to the terminator sequence, the RNA polymerase falls off. And then we have the pre-mRNA transcript that is shown here in purple. That's going to come off from the DNA as well. And as I mentioned to you already, this is not ready to go out into the cytoplasm just yet. Okay, we need to modify it a little bit to make it more stable and to edit the information a little bit before we can use it. Okay, but we'll talk about that later. So let's go back to the slides that we missed. All right. I already explained to you that we have two strands of DNA. Okay, one strand will contain the coding information, and you can't tell which one is which. Like I have to give you that information on the test. I have to tell you, oh, this one is the coding strand, this one is the template strand. You wouldn't be able to tell just by looking at the sequence. Okay, so the coding strand is the one that contains the instruction to make the protein. This is the one. This is the one that we need to make a copy of. And to build that copy, we will be using the template strand. Right? Once again, I cannot make the coding strand using the coding strand because the enzyme only knows how to use base pairing rule to make RNA. Okay, so that's why we have to use the other strand, the template strand. Hopefully you get all that. Let's do a practice question. Given the template strand, so see, I have to tell you what you're looking at. This is the template. This is the template strand. Find the coding strand and the mRNA. So let's find the coding strand. Right? So template and coding, they are both DNA, and it's easy. If you know one half, you can figure out the other half using regular DNA base pairing rule. And you can say it with me, you know, in your own room. You don't have to unmute. You can just say it out loud as I'm doing it to see if you're correct. So A pairs with T. A pairs with T. G A C. A G C. T G A T. Okay. Remember, both of these are DNA, and of course they are complementary. complementary. Okay. So now we want to make a copy of the messenger RNA. Messenger RNA. And this is the hard part. Okay? This is the part where you will show me whether you understood everything I explained to you. Okay. So why don't you type the mRNA sequence in the chat box and then press enter. Okay. Just show me your sequence. Okay, I got it from Tahib. Thank you. Anyone else? Just try it. You got to try it. Don't do it for me. Do it for you. Okay, Sebastian's got it. Have a sequence there as well. Anyone else? You will get these kind of questions on the quiz, by the way. So it's important that you know how to do it. Okay, I'm getting a few other ones. Yeah, I'll tell you if you're correct or not in a second. Yeah. Anyone else? Okay. Remember, the messenger RNA has to contain the same information as the coding strand, right? So all you have to do is just copy the coding strand. But you got to remember, the RNA has no T. So anytime you see a T, you just replace it with U. Everything else stays exactly the same. Okay? So these are identical. Identical. But the T becomes U. And not because they are base pairing rule, but because uracil is the equivalent of thymine on the RNA. You understand? Okay, so well done if you get it. It's okay if you didn't type in the chat box. If you roll it down in your notebook and you got the same sequence, very well. Let's try another one. Create a messenger RNA from the given template strand. Okay, so this is template. Template. Now, if it makes you feel better, you can do the coding first, but you don't have to. If you understand the relationship that I have been trying to show you, you know, with the picture and all that, if you have the template strand, how do you create the messenger RNA? So again, I'm going to give you a moment to type it out if you want. Thank you, Sebastian. And you know, other people, please, I encourage you to try it out. Now, if I go to the diagram I showed you earlier, right, like you can see that the bottom is the template strand, right? So if you have the template strand and you try to build a messenger RNA, you just have to use base pairing rule, right, between DNA and RNA. So in this question, A is gonna pair with U, because we're talking about RNA here, so this is just uracil. And then A, G, AAU, CAU, CGU. Questions? All right, last one. Create an mRNA from the given coding strand. Coding. This is coding strand. And I want the mRNA. It doesn't get easier than this. Okay? Remember, coding and mRNA are the exact same thing. So I just copy it, but I change the T to U. All right. If you need a cheat sheet, okay? If you're not really getting this and then you're not telling me you're not getting it, here's the cheat sheet. You can have the template strand, template, coding, and then mRNA. Right? There are only 4 possibilities, right? A, C, T, G. Okay. So on the coding, it's gonna be TGAC. Okay. What's the relationship? Complementary. What about mRNA? What's it gonna be on mRNA? Well, mRNA is same as coding, so that's, but there is no T, so UGAC. Okay. Same, but T becomes U. Okay. And what's the relationship here between the template and the mRNA? Also complementary. Okay, put that on a sticky tag, stick it on the wall, use it on the test. Okay, actually write this down because there is a 4th line that I have to show you next class. We won't have time to go to it today, but like there is another line. Okay. So I'll leave the slide, the screen on this slide. We're gonna take a quick break. We'll come back at like 1:35. Sorry, let's make it 1:40. Okay. We'll continue at 1:40. Okay, copy what you have to. If you have questions, leave it in the chat box. I'll see you in a bit. All right, let's keep going. So to summarize, the coding strand and the messenger RNA, once again, they are identical. It makes sense for them to be identical because after all, the coding strand is what contains the instruction to make the protein. So of course your RNA has to be identical to that, but with one key difference is there's no thymine on the RNA. That's why you have to replace all the thymines in the DNA with uracil. On the other hand, the template strand and the RNA strand are complementary to each other. Right? They follow the base pairing rule. And again, with a little bit of exception that A pairs with U instead of pairs with T. Okay. Let's take a look at this question. I'll let you try it first, and then I'll launch the poll. Hopefully, you guys will participate more in the poll. It's an important way for me to see if you actually understand the materials or not. Anyone else? I only had only received 5 responses. 6 now. Right? There are still 6 more of you. Choose something. This is the only way that I can tell if you are understanding it or not. Okay. 2 more students who have not answered yet. Last call. Okay. So many of you chose A. And then the second most popular answer is C. Well, what is the answer? The question gave you a template strand, and you have to find the correct mRNA sequence. What is the relationship between template and messenger RNA? Let's take a look at what the slide said. Template strand and RNA are complementary to each other. Complementary means you have to use the base pairing rule. So is not A, okay? Complementary. T pairs with A pairs with U, so on and so forth. So the answer is C. For those of you who did not choose C, have I convinced you that it's C? Do you understand why it is C? Again, moving forward is only going to get more complicated. So if you don't understand, tell me what is it that you don't understand so I can help you clarify it. Okay, template strand, RNA. You need to use the base pairing rule. Okay, different version of a gene that is called an allele. This is just definition. Genome is all the genetic information. The locus is the location of the gene, like the page number, right? I told you. Homologous pairs are chromosomes that are the same length, same size, containing same genetic information. It's like chromosome 7 with chromosome 7. Okay. Autosomes are chromosome 1 to 22. Number 23 is a sex chromosome, right? Okay, so we did all these already. And now we are gonna move on to post-transcriptional modifications. Post-transcriptional modifications. So we have our pre-mRNA now. We just made it using the RNA polymerase. But as I have been telling you, this pre-messenger RNA is not ready for to go out in the nucleus yet. It's number one, it's not very stable. If it goes out as is, it's just going to break down very quickly. And it turns out when we copy the DNA, we actually accidentally copy some non-coding information. Okay, there is some stuff that we actually don't need, and we need to take care of it. So 3 things that needs to happen. Number 1, we have to add something on the 5' end. RNA is also nucleotide, so it has directionality to it as well. 5' to 3'. We gotta add something to the 5'. We gotta add something to the 3', and then we have to do something called RNA splicing. Right. So let's take a closer look at these steps. So 5' cap and 3' poly A tail. These 2 things are going to increase the stability of the messenger RNA. It's going to make it more stable. That is important because we can change the half-life, we call it. We can change how long the messenger RNA is going to last in the cytoplasm. Sometimes we have a protein that we want to continuously make. So it makes sense for us to keep the messenger RNA for as long as possible. In other cases, we don't need the proteins for extended period of time. We just need a little bit of it, and we can stop making it afterwards. And in those cases, we can shorten the lifespan of the mRNA. Okay, so at the 5' end, you're gonna add a cap, like a cap to the bottle kind of cap, and that's made of a guanine nucleotide. Okay, ACTG, the G, the guanine. You're gonna put that in the 5' end. And then on the 3' end, you're gonna add a bunch of adenines to it. We call this the poly A tail. The more A's you add, the more stable it is going to be in the cytoplasm. So if I want to make this protein for a very long time, I should add a very long tail to it. If I just need it for a little bit, and then I don't need it anymore, then the tail can be short, right? So by changing the length of the poly A tail, I can control how long the messenger RNA is going to last in the cytoplasm. So that's the first two steps, right? 5' cap and 3' tail. And then we also have to do something called RNA splicing. Right? It turns out the DNA section that we copy, not all of it are coding DNA. Okay, there are some fillers, some junk DNA, and the junk, they are called introns. Okay, they're non-coding regions. These are junk DNA. Right? Think of like a magazine. Okay, you copy some magazine pages because you want to read the article to help with your research project, but you accidentally copy the advertisement as well. The advertisement is no use to you, so you take them out, you throw them away. Okay, before you staple together your photocopies. The advertisement are like the introns. Okay, they are junk. They don't serve any purpose in the context of making DNA. So you want to remove all of it. And then you want to glue together the exons. The exons are the coding regions of the gene. They are the sections that are going to be expressed in the proteins. Okay, so in RNA splicing, you remove all the introns, all the junk DNA, and then you connect all the exons together. All right. So post-transcriptional modification, we're going to add the 5' cap, we're going to add the poly A tail on the 3' end, remove all the introns, and then stitch together the exons. Now it is ready. Now this is the final messenger RNA that can leave the nucleus and go into the cytoplasm. All right. So this is an overview, right? We already saw the transcription that's happening here. This teardrop shape thing is the RNA polymerase. And if you pay attention, you can see the bottom strand here is the template strand. I can tell it's a template strand because the RNA is growing from it, right? And then the top here is the coding strand. After you make it, you have the pre-mRNA. There are some exons on it. That's the dark pink. That's the coding sequence. And then you have some pale pink stuff. Those are the introns. They're non-coding. And what we need to do is we remove the introns, stitch together the exons. So only dark pink. We have a 5' cap here. We have a poly A tail to increase the stability of the messenger RNA. Then it's going to exit through the nuclear pores. And now in the cytoplasm, the ribosome will come and read the information on the messenger RNA. So we're going to spend the next half an hour or so talking about the translation. Okay? This is it for transcription. We're done. But before that, let's do a few more poll questions here. The beginning of a gene is called what? Okay, good, thank you. Beginning of the gene is called promoter. Okay, initiation is the first step of transcription. If I ask you what's the beginning of transcription, then you would say initiation. But the beginning of a gene is this promoter, this AT-rich sequence. How about this one? Okay, most of you got it right. The answer is adenine. You can go back to the picture here. In the 3' end, we add a poly A tail. The longer the tail, the longer it will last in the cytoplasm. And the last one here. Okay, anyone else? Okay. The answer is intron. Okay, intron. Those are the junk DNA. Okay, let me show you the picture again to jog your memory a little bit. You see, these are the introns are the junk DNA. They don't code for anything. So we remove the introns. All right, and then we stitch together the exon. The exons are the ones that are gonna be expressed. All right, that is it for transcription. Now we're gonna move on to translation. We will not be able to finish the whole thing, but we'll start and do as much as we can. So let's do a quick review for the things that we already know. We learned this like way back, a long time ago in lecture 2, but let's go through it. DNA and RNA are nucleic acids that are made up of nucleotides. Okay, and depending if you're talking about DNA or RNA, the nucleotides will either have T or U. We know that by now. Proteins are made up of amino acids, and there are 20 different amino acids. Okay? So what we're going to learn is how exactly does the body change the ATCG into the 20 amino acids? Okay, like what's the code that allows the body to do that? So it turns out, this is like a whole chromosome right here, and then we are copying a gene only. Right? There are many genes, but we only need this one, for example. And here we have a template DNA strand. So how do we build the messenger RNA using a template? We are going to follow the base pairing rule. A pairs with U, C pairs with G, right? A pairs with U, so on and so forth. So we end up having our messenger RNA. It turns out each triplet, every 3 codes, every 3 nucleotide is called a codon. And each codon codes for an amino acid. For example, the codon UGG codes for TRP, tryptophan. UUU codes for phenylalanine. and GGU codes for glycine, and UCA codes for serine. And so each triplet, each codon corresponds to a particular amino acid. So I want to ask you, how many codons do we have? How many different codons can we create? I can create another one, like AAA, that's another one. AAC, that's another one, right? So if you do all the different combinations, how many codons are possible? Okay, we have 20 from Abnet. Anyone else? Do you guys understand my question? How many different codes are possible? You can count them. AAA, AAC, AAG, AAT. Right? No, just one answer? Okay. Well, let me ask you in a different way. How many possibility for the first blank? What can go into the first slot? Well, it could be A, C, U, or G. So four possibilities in the first blank. What about the next blank? Same thing. Could be A, C, U, or G. And the last one also 4. All right. So if I go 4 times 4 times 4, that is going to give you 64 codons, 64 different combinations. Okay. But how many amino acids do we have? How many amino acids do we have? Only 20 amino acids, right? So that means we have more codons than we have amino acids. So some of the codes are actually repetitive. Okay, they are redundant. You can have more than one code for the amino acid. Okay, and I'll show you what that means. All right, so like I said, 64 codons coding 20 amino acids. There are some repetition. It turns out the repetition is it's a protective mechanism, okay, against mutation. We'll learn about that like next class probably. So just like transcription, we need to know where to start and we need to know where to end when we make protein. So the start codon of the 64 codons, one of them is the start codon. It marks the beginning of the protein, and it's AUG. Just remember, like August, AUG. That's the start codon. Always the start of any protein that you're gonna make. And then you're gonna have a stop codon. The stop codon is the opposite. Is when you're gonna stop making the protein. And you no longer you finish the protein already. You need to stop. And there are 3 codes for it: UGA, UAA, and UAG. Okay, you have to remember these 3 codes. I mean, like you're gonna have a table to look them up, but it's much faster if you remember it because of some of the questions that we're gonna be doing. So I have a way for you to remember. It's kind of cheesy, but it works. Okay, so you got A in biology. You are awesome. You are good. Okay, so UGA, UAA, UAG. Okay, just remember that, and it will help you in a lot of the questions to make things faster for you. So you with me so far? We have the start, the AUG, and we have the end, which is either UGA, UAA, or UAG. So let's take a look at how to do a typical problem. Now you need to print out this chart to use on the test. Okay. I don't think I put, yeah, I don't think I put this in the question. because like it would take up too much space for the question and just like mess up the format. So you need to have your own table to use on the test. You cannot have a digital copy of it on the computer because you're using the lockdown browser, right? So you won't be able to pull it up during the test. So like you either print it out or you have it on your phone, okay, or tablet or whatever. Right? So that's the codon table, and I'm going to teach you how to use it. So here we have a messenger RNA sequence, right? And we are trying to change it into a protein. Okay, so we start with the start codon, the AUG, not surprising. And this is how you look it up. Okay, so A, U, look up the first letter here, A, second letter U, and then you can see where they intersect, right? They intersect in this box right here. And then we are looking for G. So over here, G. So AUG gives you the amino acid MET, methionine. You don't have to know the full name. It's just easier to say the full name sometimes. So MET, that's what it is. And I'm going to put a dash here to separate it. And let's try a second one here. This is AUU. AUU. So A, U, which means I'm in this box again. And then U, that is ILE. Okay, that's another amino acid. Let's try the next one. Type it in the chat box when you have it. Okay, let's try the next one. Okay, thank you, Sarah. Anyone else? Okay, good. Blessing. JA, good. Okay. So C is this box, and then U, that's pro. Got it? Right, and you can see all of them are pro. CCU, CCC, CCA, CCG, they are all coding for proline, the same amino acid. And that's what I was trying to tell you. We have 64 codes, 64 codons, all shown here on this table, and we only have 20 amino acids. So there has to be repetition, there has to be redundancy, right? All right, let's finish the next one here. CAU. CAU, that is HIS, histidine. Okay, I'll just give you a moment to finish the rest, and then I will write it out for you. Now you can check your answer against mine, and if you don't agree with any one of them or you want me to explain, just let me know which one. When you get to the UAA, right, you are awesome. That's one of the stop codons. So you put the word stop. Okay. Just gonna give you a moment to double check your work with mine and let me know if you need me to explain any of them. Are we good? Can I move on? All right. Thank you. Now I'm gonna, I don't know if you guys have this table with you. I give you a moment to take a picture of it. Okay, so we can do the next question. Just take a picture, take a screenshot, whatever you need to do. Okay. What does the codon CGA code for? I just need you guys to do the poll so I know all of you are getting it. Okay. Okay, I'm missing 3 answers from 3 people. Okay, choose something. If you don't see the poll, you can type the answer in the chat box instead. But I have to see it. I have to know that you can do it. Because the rest of the class and next class is gonna rely on, you know, being able to use the table. Anyone else? No? Okay, I think the other 2 people is perhaps not here. All right, most of you got it right. The answer is A. Some of you chose other stuff, so let me go through it one more time. CGA. CGA. So that's what I need to find. So C and G. Right, so C is this column, sorry, this row. G is this column, so I'm looking at this box. And A, right? So like they are all arginine. Okay? That's how you do it. This one is difficult. I'll just I'll let you read it and then I'll show you how to do it. Okay. Now, when you see a question like this, instinctively, a lot of people choose E, but that is not the answer. Like you're like, what do you mean? You said, you said you got A. That's the stop codon. Yeah, be that as it may, but this is talking about the template DNA. Right? This is talking about the template DNA, and there's definitely no uracil in the DNA. So what would we do? Right, let's write down the mRNA first. And so we have 3 stop codons, right? You got A, you are awesome, you are good. But don't forget, the messenger RNA actually came from DNA, right? So it came from copying it from the template DNA. And the relationship between the template and the RNA is that they are complementary. Right? So that's like U is going to be A on the template, CT, ATT, ATC. Right? So we're looking for this. There you go. ACT and ATT. Right? The answer is B and C. B and C. Okay, because we're looking for the template DNA. Give that a little bit more thought and think about it next time. All right. I am gonna skip this example. You can do it for practice. We'll take it up next time, first thing on Thursday. But I want to do this one with you before we say goodbye. So this is the from the beginning, right? We are given the template strand, so we have to do transcription first. Transcription to get the messenger RNA. So you got to be very careful on the quiz. Because if you get your messenger RNA wrong, then your protein is going to be wrong as well. Right? So you have to be very, very careful. So what is it? Right. Well, the template and the messenger RNA, hopefully you know by this point, they are complementary. Right? So TAC is going to become AUG. And I'm just going to put like a dash here to space them out so I can see it easier. GTA is going to be CAU. Please check my work. Sometimes I do this thing and then, you know, I misread a letter or something. AAT will be UUA. ACA is UGU. CCG, UGA, like that. We will have chance to practice next time together. Okay? Of course, you should practice, you know, on your own as well, but that's what it is. There are more questions in the study guide. And then we're gonna have the protein here. We're just gonna look them up. AUG is MET. CAU GGA UUA. You can watch the recording afterwards to see how I'm doing it, right? UGU CCG, and you got A. That is a stop codon. It says stop right here, right? You see? That's it. That's how the body does it. It performs the transcription, and then I actually should put an arrow here that says translation. But the ribosome is gonna essentially read the nucleotides in triplets and then find the corresponding amino acid. Okay. So that is it. We're gonna finish the rest of lecture 7 next class. And you will need the study guide. I know I told you to print it, but we didn't end up using it just yet. So you know, make sure you print that for next class. We will build upon this thing next time, and I hopefully we'll have some time to talk about the lab as well, because your lab is happening next week, and you also have the quiz next week as well. I think. Let me just double check. Yeah, we're here right now. So you will have the quiz on Tuesday, and then you will be doing your lab next week as well. So I'll tell you more about it probably next class. It's a very simple lab. Okay, like there is a simulation that you have to do on the microscope, and yeah, okay, we'll see how it goes. All right, thank you. See you all on Thursday. And if you need extra help with this, you know, CAG, TTC stuff, just send me an email and I'll help you out. Have a good one. Bye-bye.